Hi guys, let's do section 2, Mendelian genetics. So, the essential questions: what is the significance of Mendel's experiments to the study of genetics? What is the law of segregation and the law of independent assortment? And what are the possible offspring from a cross using a Punnett square? So hopefully by the end of this unit, or of this chapter, of this section, there we go, you should be able to answer those 3 questions. Here is the vocabulary that you had a test on earlier this week. If you still have not written them down, take a few moments and do that. How genetics begin. The passing of traits to the next generation is called inheritance or heredity. Gregor Mendel was the first person to begin studying this, and he published his findings on the method of inheritance in garden peas. He used garden peas in his studies because they have lots of different traits that he could study, and they did not have intermediate traits. So one of the traits that he studied was plant height. So if he crossed a tall plant with a short plant, they did not make a medium plant. Okay, it was either tall or short. There was no in between, which made his research much easier, even though it was still very complicated. He rigorously followed the traits, like the height of the plant or the color of the seed, through many generations. So he didn't just look at the seeds from the plants. Okay, he looked at several generations. So he planted some pea plants, and then they grew, and he got some seeds, and then he planted those seeds, and he looked at the traits that those plants had, and then he planted the seeds from those, and over and over and over. And thus began the study of genetics, which is how traits are inherited from one generation to the next. The inheritance of traits. One trait that Mendel noticed was seed color. Some plants always produced green seeds, while some always produced yellow seeds. So Mendel crossbred the green and the yellow seeded plants. He did that by removing the male sexual organs, the stigma and the style where the pollen is found from certain pea plants, and he took pollen from another pea plant and fertilized them or pollinated them. And so he cross-pollinated them. That's what that process is called. And he called those parental seeds, those green seeds and those yellow seeds, the parent or pea generation. Okay, so those were the parents, the pure breeding yellow pea. Okay, so if he let it self-fertilize, it would only produce yellow peas. And the green pea, if he allowed it to self-fertilize, it would only produce green peas. So he bred those two together, and that resulted in the first filial generation, or the F1. And he noticed that all of the offspring were yellow. He then let those yellow seeds self-fertilize. He did not remove the pollen-producing structures of the flowers. He let them reproduce the way that they would normally. And the second filial generation, the F2, or the offspring of the parents, produced yellow and green seeds. All right, so the offspring of the pea cross are called the first filial generation. The second filial generation is the offspring from the first F1 generation. In Mendel's peas, the green-seeded trait disappeared in the F1 generation. There were absolutely no green seeds in the F1 generation, but they reappeared in the F2 generation. And when Mendel looked at the number of yellow seeds compared to green seeds, he noticed that there was a 3-to-1 ratio. Okay, so if he had 100 seeds, approximately 75 of them would be yellow and 25 of them would be green. That's a 3-to-1 ratio. Mendel studied 7 different traits of pea plants. He studied the seed or the pea color, the flower color. The flower color could either be white or purple. The seed pod color could be either yellow or green. The seed shape or the texture, they could either be round, a perfect circle, a perfect sphere, or wrinkled, kind of bumpy and wrinkled in texture. Seed pod shape, they could be inflated or bumpy, I think were the terms that he used. Stem length, also called height of the plant, was it tall or short? And the flower position. It is terminal or axial. Terminal means that the flower is at the very top of the plant, and axial means that it is about halfway up. In all cases, when Mendel looked at one of these traits, he always found that the F2 generation showed a 3-to-1 ratio. Genes in pairs. Mendel concluded that there must be 2 forms of the seed trait in the pea plants. Okay, so there's 2 forms of each one of these traits listed. So the flower color could either be purple or white. Okay, there were 2 different forms, and that each was controlled by a factor. An allele is an alternative form of a single gene. So green or yellow, purple or white, tall or short. The gene for yellow seeds and the gene for green seeds are different alleles for the same gene. So they control the same thing, they just look different. Dominant alleles control the traits that appeared in the F1 generation, and recessive alleles were masked in the F1 generation. Recessive alleles can be present, but they can be masked by a dominant allele if the dominant allele is there. If there's no dominant allele, there's no masking it, and the recessive allele will be shown. Dominance. When modeling inheritance, the dominant allele is represented by a capital letter, in this case a capital Y, and a recessive allele is represented with a lowercase letter, in this case a lowercase y. An organism with 2 of the same alleles for a particular trait is said to be homozygous for that trait. So it would be capital Y, or lowercase y. Big Y would be homozygous dominant because dominant traits are represented with a capital letter, and lowercase y would be homozygous recessive because homo means same, so it's the same, recessive means lowercase. An organism with 2 different alleles for a particular trait is heterozygous for that trait, so we would have a capital Y and a lowercase y. In heterozygous individuals, the dominant trait will be observed because it will mask, it will overshadow, it will overpower that recessive allele. So the recessive allele will not be shown, but the dominant one will. Genotype and phenotype. The appearance of an organism does not always indicate which pair of alleles it possesses. So you can see the dominant look, but you might not know if it's homozygous dominant or heterozygous. An organism's allele pair is called its genotype because the gene controls the allele. The observable characteristic or the outward expression of an allele is called its phenotype. I always think phenotype is the physical appearance, and genotype is controlled by its genes. Okay, so phenotype I can physically see. Genotype I have to know the alleles that are present. The genotype controls the phenotype because genes control what it looks like. Now let's move on to Mendel's law of segregation. The law of segregation states that the 2 alleles for each trait separate during meiosis. Remember, 1 chromosome goes to either side of the cell. Okay, you only inherit 1 chromosome for each trait. Okay, you don't get both pairs from both parents. You only get one or the other. During fertilization, those 2 alleles for that trait unite. So you get 1 from the sperm and 1 for the egg. Heterozygous organisms are called hybrids because they contain both alleles. A monohybrid cross is a cross that involves hybrids for a single trait. Okay, so you have 1 single trait that you are looking at in a monohybrid cross. Mono means 1. Di means 2. A dihybrid cross is when you are studying the outcome of more than 1 trait. So the presence of a round seed and a yellow seed, or a wrinkled seed and a green seed. So you're looking at 2 different traits. Okay, 2 different traits in a dihybrid cross. The law of independent assortment. The law of independent assortment states that random distribution of alleles occurs during gamete formation. Basically, that means that Just because I have a dominant color does not mean I'm going to have a dominant shape. Okay, they separate independently of each other. Genes on separate chromosomes sort independently during meiosis, and each allele combination is equally likely to occur. So there is a 1 in 4 chance that if I cross a yellow and, if I have a yellow and a round, okay, that my offspring is going to be yellow and round, because those traits are possible from my parental cell. But it's also equally likely that I'm going to have a green and round. Okay, it is equally likely that I'm going to have a green and wrinkled, or I'm going to have a yellow and wrinkled. There is a 1 in 4 chance of getting every single one of those combinations. Punnett squares are an easy way to figure out what possible genotypes you could get from crossing two individuals. Punnett squares predict the possible offspring of a cross between two known genotypes. You have to know the genotype of the parent to be able to predict the genotype of the offspring. Okay, and it's not guaranteed 100% of the time. It's a probability thing. And each time that the parents reproduce, there's the same chance that you're going to get a tall plant or a short plant. So in a monohybrid cross, the number of squares is determined by the number of different types of alleles produced by each parent. So each combination, if I only have two alleles that I'm looking at, so I'm looking at one single trait, I'm going to separate them. I'm going to put a big T here and a little t here. Then I'm going to look at my other parent, and I'm going to put a big T and a little t, and then just like a multiplication table, I go up and I go over. So I have big T here. If I move over to the next one, I have big T, little t. If I move on, big T here, little t here. So I have big T, little t. And if I go here, I have a little t, so I'm going to have two lowercase t's in this box. Don't worry, we're going to do lots of practice with this. A dihybrid cross is when you are looking at two different traits and wanting to know the chances that they're going to inherit those two traits at the same time. So you would end up with 4 boxes across the top and 4 down the side. So you would end up with 16 boxes all together. Okay, I just want you to know that this is a thing that exists. If you want to try a dihybrid cross, you can. It will be bonus points, but I'm not going to require you to know the dihybrid cross. We're going to focus mainly on monohybrid crosses. If we cross two parents that were dominant for both traits, and we get heterozygous for both traits, and then we cross those in our F2 generation, we're going to end up with 9 seeds that are yellow and round, 3 seeds that are yellow and wrinkled, 3 seeds that are green and round, and 1 that is green and wrinkled. Okay, it's a phenotypic ratio. The number here needs to add up to the total number of boxes that you have over here. So 9 plus 3 plus 3 plus 1 is 16, and that matches up with the number of boxes over here. Probability. The inheritance of genes can be compared to the probability of flipping a coin. The actual data might not perfectly match the predicted ratio. Mendel's results were not exactly a 9:3 to 3:1 ratio, but the larger the number of offspring involved, the more likely it will match the results predicted by the Punnett square. Each time I flip a coin, there's a 50-50 shot that it's either going to be heads or tails. If I flip a coin 50 times, probability says that I should get 25 heads and 25 tails, but that's not always the case. Sometimes I get 60 heads and 40 tails. But if I do it enough, if I flip the coin enough times, I should get a 50-50 ratio. And that's it, guys. If you have any questions, let me know.